Yes.
Perfect. Okay, so we're going to start off by reviewing parts of the microscope. So for this one, I am assuming that everyone is familiar with microscope parts. So you guys should be able to just shout out the names or write them out. So let's start from the top. What are these lenses on the top called? Ocular lenses. Good job. So yeah, so those are your ocular lenses. You also call them eyepieces. So that can be your eyepiece as well, since that's where you look at it. In this case, they will be binoculars, since there are two lenses. Sometimes you only have a single lens, and sometimes you have two. Obviously, it's much more comfortable with the two. With these, you can increase or decrease the distance between the two lenses to your comfort and to your measurements. So that is good. Now, these lenses themselves magnify your image anywhere from 5 to 20 fold. Um, and that's important in figuring out your final magnification of images when you're looking at them under the compound microscope. So the ones that we use in the lab are 10x, and so that's the one that we are going to be looking at. All the measurements that I give you, we are going to assume that the ocular lenses are 10x magnification, and that's what they have. Okay, now going underneath, what is this revolving piece called? We have this rotating piece underneath the ocular lenses. What are those called? What is that called, rather? Yep, it's the revolving nose piece, and it contains what? So there are four different lenses. What are they called? The objective lenses. That's right. Good job. Now there are four different objective lenses, and they are going to range in anywhere from 4x all the way up to 1,000 or even 2,000x, depending on the microscope that you look at. Um, so the microscope that I have, had 4x objective lens, or most of the ones that we are going to be looking at, they will have 4x objective lens, which is the smallest one that we will see. And then the highest one that they will usually have is the 100x objective lens, which is the highest magnification that you'll get out of these. Now, depending upon what you have up here, if you have a 10x objective lens as your oculars, then the maximum magnification that you can do with this microscope would be what? So remember, the highest objective lens here is 100. So what would be the maximum magnification that you can do with this microscope, given that your highest objective lens has 100x magnifying power? Yep, good job. Would be 1,000x. And what would be the minimum magnification that you can do? Considering that the lowest objective lens has 4x power. 40x, that's right, good job. Now, these lenses have different names based on their qualities and based on what they are used for. What is the smallest magnification lens called? So the 4x objective lens. Anyone remember the name for it? Scanning objective lens, good job. So yes, so the lowest objective lens is called the scanning objective lens. The reason for that is that when we are looking at this specimen underneath the microscope, this will allow us to see the maximum area of that slide. And so we can use it to scan our specimen and pick the area that we care about, pick the area that we want to focus on. And then we can magnify the area of interest further to look at it. Next, we have low power objective lens, right? And our low power objective lens can be 10x or 20x power. Usually the ones we have in our lab are 10x objective lenses. And then we have high power objective lens, which can be 40 or 60x. And the ones we had in our labs were 40x objective lens. The images you're going to use today are going to be taken using the 40x objective lens. So other than Benton, somebody else, what would be the final magnification of the images that you will receive today for analysis? So remember, it's 40x objective lens and 10x ocular lens. So 100x is not correct. No, try again. 400x. Good job. Benton, I thought I told you not to respond. Well, that's okay. You're on probation. No more responses for 5 minutes. Okay, so we got all our lenses. Next, let's go down further. We have our stage. This is where we're going to keep our slide. Our stages are usually equipped with some sliding rulers, so you can move the slide either left to right or front to back as needed to get the specimen in on, to get the specimen focused on the area that you want to look at, that you want to work with. So you just move it around using those knobs and not actually physically touching the slide. You want to always minimize your actual hand manipulation of the slide, because that can put fingerprints all over them, make them dirty, mess them up, squish the specimen inside, and just, destroy basically your observation power. So you don't want to do that. Now, once we have placed our specimen onto the slide, we need to focus it. To focus it, there are two knobs on the handle of the microscope. What is the inner larger knob called? It's the coarse focus, exactly. So it's the coarse focus knob. Now you can only use the coarse focus when you are looking at the microscope, at the specimen using the scanning objective lens. That's its job, is to focus, give you a general focus of your slide, and then you use the fine focus knob, which is the inner smaller knob, to fine tune your focus and bring it into the resolution you want. Once that is done and you move from your scanning objective lens to your next objective lens, the only knob you should use is your fine tuning knob to fine tune your focus. You should not have to use the coarse focus. If you're having to use the coarse focus, you should be going back to the scanning objective lens and restarting to make it focused initially. Now, let's say sometimes we get samples that are really hard to focus and you're having a hard time with them, or you're unable to find them. Well, what you need to do is go to the scanning objective lens and move your stage all the way down to the bottommost level it can go, and then restart from that point. Because at that point, there is no wrong way. The only way you are going to find your specimen is to go up. And so it takes away your kind of, confusion about whether you're going the right way or not. And eventually, if you go up, you will find your specimen. Okay, now, specimens that we will look at, some of them are going to be stained, obviously, with different dyes, so you can see the cells and the inside of the, the organelles a little bit better. Other times, you're just looking at bright field images where you don't actually have any stains. The cells are the way they are. Now, most of the cells that we're going to be looking at are going to be pretty translucent, right? So it's like clear on clear. In that case, what are some things you can do to enhance your contrast between the background and your actual cells so that you can look at them and observe them better? What things you can adjust on the microscope to help you with contrast? Yes, you can adjust the amount of light. Okay, so now let's think about ways that we can adjust the amount of light. And on that note, actually, would you want your light to be higher or brighter, or would you want it to be lower in order to enhance contrast? If you do it higher, what you will find is that sometimes you don't even see your cells. So we do this counting small objects lab where we count the number of yeast or number of cells in our sample. And if students forgot to adjust light, they would be like, I don't see anything. My partner sees thousands of them. I don't see a single one. And all I would do is go to their microscope and move one little switch, and they would be like, oh, there they are. So you don't want to adjust them higher. You want to adjust the light to lower setting. And in general, it is good practice to keep the light as dim as possible while observing your samples, because it's better for your eyes as well. So especially, I've been in lab, I've done experiments where I'm looking under a microscope for 12, 14 hours straight. If I had the light adjusted super high, my eyes would be shot, right? So instead of that, you always want to adjust the light towards the lower side. But it's especially important to do that when you're looking at images or actual cells that are translucent, that are not stained. Now, obviously, we can adjust the light with just this, with the actual light intensity over here. So you can adjust the brightness from the light source. But there's another way that you can adjust it. What is that? There's one other way to adjust the amount of light. Yep. So it's the aperture, and you can control it with the condenser and the diaphragm setting down here. So there's a sliding switch that goes underneath that adjusts the amount of light that is coming to your specimen. And I can make that amount of light. So you are adjusted the brightness of light from the actual light source, and you can make it as dim as possible. And then you can adjust the amount of light that's actually coming to your sample. And you do that by moving that switch left to right, depending on how you want it. So that's what I usually did, was that I would just go by the student and switch over the aperture to the lowest setting, so less amount of light was coming through, and lo and behold, cells would appear. So that's the other thing that you can do. Good job. So that's our general microscope. We got that. Now, obviously, we use the microscope to magnify our images, our specimens, so we can observe them, so we can make qualitative observations about them, about their shape, about the way they are behaving, about their physical characteristics that are changing in response to drugs, in response to environmental stimuli, whatever it is that we are looking at. Now, there are quantitative measurements we can take from the microscope as well. However, they involve, calibrating our microscope so that we can do that. The reason for that is that if I were to look at a specimen, let's say that I'm looking at a cell of whatever type underneath the microscope, some mammalian cell. Let me switch on. Oh, no, 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 no. One second. There was a little, I want to get my pen out from here. Okay. So let's say that I have a little cell and I'm looking at it under 4x objective lens and I see, some dot and I see a bunch of them around it. So I know that that's my cell. I move over to the 10x objective lens and I can see a little bit more detail in there. And I can see a maybe tiny nucleus inside. And I'm like, oh yeah, it looks like a animal cell, right? And then I move over to my 40x objective lens and now I can see more detail in there, right? See the nucleus, I can see the nucleoli, right? Maybe I see some cilia, who knows, something else is there. Mine used to be a touch screen, but I dropped it a few times, so now it's cracked, and there are portions of it that are touch screen and portions of it that are no longer touch screen. And then I see, maybe little mitochondria in there. Who knows? Maybe not at this one, but at 1000x definitely. I'll see a few more things. So I see a lot of details now, right? Now, if I was to try and figure out the size of this cell, I can't just simply do that looking at this, because obviously the cell is still the same size, right? If the cell was, or thinking about me as a human being, if I'm 5 feet up here in my room, I'm also 5 feet downstairs in the yard, and I'm 5 feet at UNF, and I'm 5 feet in, wherever I am in the world. So my size doesn't change just because I change my environment. Similarly, their size, the size of the cell doesn't change simply because I'm looking at a different magnification. But yes, I am magnifying it. So what I need to do is have a ruler that I can compare it to and then actually decide what the size is. So in our ocular lenses, in one of our ocular lenses in the lab, we have a little ruler. And that ruler is called an ocular micrometer. That ruler has, 10, well, the ruler is subdivided into 100 tick marks, okay? So I'm just making the big ones, not the small ones here, and obviously not a very good one either. And so, so I made the big 10, and then each one of these is subdivided into 10 parts like this. So you have, this ruler is divided into 100 little subunits. Each one of those is called an ocular unit. Okay. Now, when I compare the size of these little cells, the cell at different magnification, maybe it's going to be 1 ocular unit over here, right? And then, no, yeah. And maybe then it is 10 ocular units under 10x. I can't write with my mouse, it's sad. So the size of my cell is going to look like it is changing a lot, right? And maybe it is 25 ocular units here for all I know, right? So obviously my cell is not suddenly changing into size, but the magnification is changing. What I need is a ruler of known length, something that I know how long it is, that I can put on the stage, stage, just like my slide, and see how that magnifies and how that size changes. So we do that by using a slide that has a ruler on it before we look at our specimens. And that ruler is called a stage micrometer. That ruler is also subdivided into 100 tick marks. Each one of them is called a stage unit. And what we do is we use the knowledge of its length in each one of the stage units, and we create a particular, essentially a conversion factor between the stage unit and the length. So we know how many, what length each stage unit is worth, and we compare it to how many ocular unit it compares to at each magnification. And that's the whole idea behind the calibration, okay? So far so good or no? Yes, no, maybe. So we're gonna go through that calculation in a second. How do I do this? Why won't it let me? Go to the next one. Apparently. Oh, I have to delete this or do what to the? No, 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 no. Okay. So let's go to the next one. Come on. Okay. So the stage micrometer has a set length as far as itself, right? It's going to be different for every single microscope. So that's why we have to, every single person has to calibrate their microscopes. But the stage micrometer is going to be a given length. It's on a slide. And so when we look at it under the microscope, we know what that length is, and we can compare it to the length of the ocular micrometer of that microscope, and then make our calculations for it. You'll see once we go through a set of calculations how that works, okay? So if we were in the lab, we would be first asking you to look at that ocular micrometer, so the one in the eyepiece, right? And I always tell students to go ahead and switch their nose piece lenses, so their objective lenses, and look at how it changes the ocular micrometer. So in our case, you don't have your microscopes there, but think about that. You have your ocular lenses on the top, right? Then you have your nose piece and the objective lenses, and then you have your stage. So what I'm asking you to do is to take your look at that ocular micrometer, so the one on the top, which has a ruler in there. That ruler doesn't, we don't know how long it is right now, but it's there, and it says 100. It goes 10, 20, 30, 40, all the way to 100. And you're going to change the magnification of your specimens using the nose piece. As you go from 4x to 10x to 48x objective lenses, would you expect the size of the ruler in the ocular lens to change? So just write yes or no. Okay. Anyone else? Apparently 30% of the people vote here, right? Okay, so no, it won't. Why not? Why wouldn't it change? Anyone want to take a stab at it? Why wouldn't the ocular micrometer change when we look at it under various magnifications here? That's right. So the ocular lens is up here. That's where the ocular micrometer is. If I change the objective lenses down here to various magnifications, there's nothing underneath it. It's my specimen is underneath it, but that ocular micrometer is above it. So you will not get any change in that. That ruler is going to look exactly the same no matter which magnification you're focusing on. So that ruler does not change. However, if I were to put a slide containing a ruler on the stage, and then I change the magnification using the nose piece objective lenses, that one is going to change because that will be below the objective lenses, right? So if I have a slide with a ruler here and I change this, I will see it grow bigger and bigger or smaller and smaller as I go from one magnification to the next. So that's what we are going to do. The ruler that we use is a 1 millimeter ruler. How many micrometer would that be? So 1 millimeter ruler, how many micrometer would that be? A little bit off. No, more off. We're getting colder. 1,000, good job. So yes, 1 millimeter is equal to 1,000 micrometer. Now we want to think in micrometer terms because cells are small, right? We're looking at molecular things. They're going to be tiny. And so 1 millimeter cell is going to be hard to find, but you may find cells that are 2 microns or micrometer or 10 or 20 or 40. So that's what we're going to be looking at is in micrometer. So we have ruler that is the stage micrometer. It's also going to be subdivided the same way. It won't have any numbers on it, but that's okay. We don't need them. We know the length. It's a 1 millimeter total or 1,000 micrometer total. So if there are 100 subunits, 100 stage units in the stage micrometer, how many micrometer is each stage unit? So 1,000 micrometer in the entire length and 100 stage units in the entire length. So each stage unit is going to be worth how many micrometers? 10. Good job. You got it. So what we're going to do is know that that is the conversion factor, that each stage unit is going to be equal to 10 micrometer when we start looking at calculation. And that conversion is true regardless of the magnification. No matter which magnification we are looking at, one stage unit is going to always equal 10 micrometer. Okay, since that is a known information. So this is just going over what we just went over, that, you know, the total length of the stage micrometer is 1,000 micrometer, and one stage micrometer is going to be 10 micrometer. So what we do next is that we calibrate it to the easiest one, which is the 10x objective lens. So these pictures are from our microscopes at work. So, you know, I accidentally got a not so good microscope, so it's a little bit dirty, but that's okay. We can still see. So what we do is we line up, we make the image horizontal, we make our ocular lenses, micrometers, so our little ruler in the ocular lens horizontal in our field of view, so we can look at it easily. We then take our stage micrometer, so our slide, and we bring it up and focus it so that once it is focused and I can see both the stage micrometer and the ocular micrometer. And two, I move the slide so that I line up both of them at their beginning. So I line them right on top of each other and starting at the 0 time point or 0 point of those rulers. So when I do that, I can look at them and I can see how they fit against each other. So at 100x total magnification, so this is looking at a 10x objective lens, we look at which one is bigger or smaller than the other. In this case, it just so happens that they both line up exactly on top of each other. So the 100 ocular units are right where the 100 stage units are. So what we do is we write that down. Okay, so in this case, we make that conversion and we say that 100 stage units are equal 1 millimeter and 1,000 microns. We already did that. And that means that each stage unit is equal to 10 micron. We then make an equation where we take 100 stage units and equal them to whatever ocular units they correspond to. In this case, the 100 stage units were equal to 100 ocular units. So they were both exactly the same. So that's all good, but that doesn't help me, that doesn't give me an actual measurement to go by. To do that, I have to convert the stage units into an actual unit of measurement, which is micrometer in our case. So I multiply it by the conversion factor that I know. So 100 stage units, where each stage unit is equal to 10 micrometer. I multiply by that conversion factor. The stage units are going to get canceled out, and you will get 1,000 microns is equal to 100 ocular units. And then same way as we did up here, we divide both sides by 100 so that we can find out what each ocular unit is going to equal to. And in this case, it is going to equal 10 micrometer. Okay. Oh, let me, yeah, that's fine. So this is for our 10x objective lens. This would be our calibration for that lens on that microscope. So if I have a cell, right, let's go back and get a cell. If I have a cell and under the 10x objective lens, it is 2.5 ocular units, okay? Then how many micrometer is that? My corrects are showing up. How many micrometer would that one be? Remember, each ocular unit is equal to 10 micrometer under this lens. So if I have a cell that is 2.5 micrometer, 2.5 ocular units, how many micrometer would that cell be? So to figure that out, all you need to do is look at how many ocular units you have and multiply them by the calibration, which is 10 micrometer. Good. So the answer would be 25. So if your cell is 2.5 ocular units long, then it is 25 micrometers under the 10x objective lens. Okay? Now, if your cell is 25 micrometer under the 10x objective lens, how long should it be under 4x or 400x or 1000x magnification? As far as the micrometers are concerned, what should be the length of that cell under any other objective lens? 25. Good job. So keep that in mind. Same thing as what I said before. I am 5 feet no matter where I go. I can go to Mount Everest, I'll still be 5 feet. I will not increase in height. Similarly, this cell is going to be 25 micrometer no matter where he is, under what magnification he is. But its length in ocular units will change corresponding to the magnification. So let's do one by ourselves. So I'll give you what it looks like. So under the 4x objective lens, okay, so while we are at the 4x objective lens, which is the 40x final magnification, this is my ocular micrometer. So this is the full length, okay? So this is 0, this is 100. And here is my stage micrometer. Okay? So this is my stage micrometer. And this is going to be 4x objective lens. So under 4x objective lens, when I look at them, I still see the entire length of both micrometers, both the ocular and the stage. But now I only, one of them is super tiny, right? The stage micrometer, because we're on the scanning objective lens, is very small. And the ocular micrometer looks the same as it did before. So now what I need to do is draw an imaginary line from the end of the smaller one to the other one and see what it corresponds to. So what should I write for my equation in this case? So before they were the same. So I wrote 100 stage units are equal to 100 ocular units. Now they're not the same. So what would I write for my equation? How many stage units are going to equal how many ocular units? Anyone? 100 stage units is good. So yes, it is 100 stage units because you see the entire length of the stage units or stage micrometer. However, it is not 5, but rather 10, 20, 30, 40, right? So it is 40, 10, 20, 30, 40. So it's going to be 40 ocular units. That's okay. So go ahead and work out the math exactly the same way. You're going to convert the stage units to micrometer using the same conversion factor. And then you are going to solve it out for 1 ocular unit. So your final answer is going to be in, micrometer per 1 ocular unit. What would it be? You're doing it just the way that we've done it before. So you're going to say 100 stage units multiplied by 10 micrometer per 1 stage unit equals, instead of 100, now it will say 40 ocular units. Then you will get the same 1000 micrometer, but this time those 1000 micrometers are going to be equal to 100, instead of 100, 40 ocular units, right? So it will be 1,000 micrometer equals 40 ocular units. You're going to divide both sides by 40, so you will get, what will you get? Nobody tells me. So sad. Good job. 25 micrometer per a single ocular unit. Same cell. Remember the cell? Now I'm looking at it under the 4x objective lens. How many ocular units is it going to be? So remember that cell was 2.5 ocular units under the 10x objective lens. And the total length of the cell is 25 micrometer. How many ocular units is it going to be under 4x objective lens? No way. That would be huge. You don't want it to be 62 point. Why would it be 62.5? Doesn't make sense. I don't know. How did you calculate that? Try again. No, you don't want to do 25 times 2.5. Remember, this it's 25 divided by 25 actually. The total length of the cell is 25. That's what you want to get to, is 25 micrometer. In this case, each ocular unit was equal to 10. So you divide that by 10 and you get the 2.5 ocular units, which is what you saw. This time, it's still the same 25. You divide it by 25 and you get 1. So it's going to be 1 ocular unit. And that's how big the cell is going to be. This tiny little blob. That was not done very well. So it's just going to be a tiny blob with a tiny speck inside. Okay, you guys get that? Okay, we're going to do one last one because it changes things up a little bit before we go to image J. So now we are going up to the 40x objective lens. And this is when things get interesting. So now you still see the entire length of the ocular micrometer just like you did before. That never changed, right? So that didn't work. How do I make it go away? Who knows? Well, it's there, right? And this is always going to look the same no matter what magnification you're looking at. But now you only see a portion of your stage micrometer because you have magnified it 400 times. So when you're doing your equation this time, you will see the entire length of your ocular micrometer. So all 100 ocular units, but you're only going to be seeing 10, 20, 1, 2, 3, 4, 5, 25 stage units. Okay? Now you solve it out just like you did before and tell me how big this, what the calibration is going to be under the 40x objective lens. 1 ocular unit is going to equal how many micrometers? So just like before, you're going to take your stage units, you're going to convert them into micrometer using the same calibration, 10 micrometer per stage units. And then you're going to divide both sides by 100 to get how many micrometers per ocular unit you will find. So what would be the calibration? First of all, so it should be 250, right? Once you calculate, it's 25 times 10. So it's going to be 250 micrometer is equal to 100 ocular units. Then you need to divide both sides by 100 before you get the final answer. So it would be 2.5. Good job. So yes, 2.5 would be the answer for this calibration. So this is going to be your calibration for 40x. And then same cell. Remember that cell? That cell was 1 ocular units under 4x objective lens, was 2.5 ocular units under 10x objective lens. How many ocular units is it going to be under 400x or 40x objective lens? If you remember, it's 25 microns in length from before, right? So how many ocular units is it going to be? The same way, if you know the length of a cell, you can just simply divide it by the calibration to know how many ocular units it's going to be. So at 25 microns and 2.5 micrometer per micron, right? 25 micrometer is the total length, and 2.5 micrometer is length per ocular unit. How many ocular units is it going to be? Not 2.5, but 10. I don't know how you got 12 and a half. So it's going to be 10 ocular units because it's 25 divided by 2.5. And now you should be able to see a lot more in that cell at that magnification. Cool? Okay. So this is just showing you that, again, that it was 25 stage units, and then 250, and then 2.5. And this is just a review of that. And these are the numbers that we will use for the rest of the semester anytime we are going to be looking at a microscopic slide. But this is only if we are looking at images without image analysis. Image analysis will have its own calibration. So under 40x total magnification, 1 unit was equal to 25 micrometers. At 100x total magnification, which is the 10x objective lens, 1 unit was equal to 10 micrometers. And under 400x, which is what we just did, it was equal to 2.5. We didn't do the one for 1,000, but at 1,000, it's equal to 1. And these were the numbers we would have used in the lab on our actual microscopes. Now, just like we talked about right here with this imaginary cell, if I have two different objective lenses and their respective calibrations, right, and I measure the same blood cell, right, the same exact blood cell, I look at it under two different magnifications and I calculate its size, is it going to give me the same size or different size? These are all good quiz questions, by the way. Same size. Good job. And here are just examples of how different they look under these objective lenses. So you can see from very little information to a lot of information from these cells as we go from the 40x scanning objective lens to the high power 400x.